Okay, so we're going to go ahead and get started. Scott, could you just drop the link to the slides one more time in the chat, please? The slides I'm going to be showing you today are an overview. They are not meant to be exhaustive to describe every single thing that is written in Unit 2, Biomolecules of Patterns Biology. However, what I've tried to do here is to provide you links to documents that are a little bit more exhaustive so that if you want a more detailed picture, then you are able to see that relatively easily. So I think it's important to start because some of us have more access to or more familiar with using patterns. Some people have 5, 6, 7, 8 years using materials from patterns. And then some of us also have experience teaching different courses within the pattern sequence. But some of us are relatively new to patterns. And so I think it's important for everybody to be aware or remind themselves that the patterns curricula are devised and designed with certain principles in mind. And we call these the design principles. And you see them listed there on the left-hand side of your screen. I will say that we do have details about those in 2 documents. One of them is a 2-pager, which is kind of a condensed version. And then you have the full version there, which I think is probably double that length. And I think that I share this document or this information with you because I think it's important for users to understand the kind of why behind the decision-making process of what goes into a patterns unit or what kinds of activities are selected to be part of a patterns unit. Also why we do inquiry and engineering the way that we do. So we don't have time today to go into those in detail, but I wanted to make sure that you have access to those so that you can peruse them at your leisure and to have a little bit more context about the development of patterns curriculum. So today we are going to be working on the Unit 2 of biology. Patterns biology is taught when the whole sequence is done, it's taught to 11th graders. I did see some people in the Jamboard wrote in that they are using this curriculum for 9th graders, but needing to make some adjustments. And certainly I can understand why that might be necessary if your district teaches biology to 9th graders. But in this unit, we're answering a couple of essential questions. First of all, how do cells make the molecules they need and get rid of the ones that they don't? And what happens if steps in this process aren't working correctly? And we are addressing 2 NGSS performance expectations in this unit, the biomolecules performance expectation and also DNA to protein. So this is a way in which patterns biology is a little bit different than there are lots of ways in which patterns biology is different from a traditional biology curriculum, but this is one of them in that we're not teaching DNA and protein or DNA and protein synthesis in a separate chapter. We're addressing that because DNA and proteins are biomolecules, and so we're addressing them within the unit around biomolecules. So if you are coming from a more traditional teaching sequence, then you may be surprised to know that we're not teaching DNA in 2nd semester. We're teaching it almost at the beginning of the year. And our anchoring phenomenon has to do with baby Matthew. So we'll talk quite a fair bit about baby Matthew today, but our phenomenon has to do with baby Matthew is born and seems healthy in his first few days of life, but then he starts to exhibit some strange symptoms and his parents take him to the hospital and are wondering what is happening with their baby. They're very concerned. And the phenomenon allows students to help try and figure out by the end what's happening with baby Matthew. And spoiler alert, baby Matthew is able to be saved. So that's good. So some helpful resources for you to navigate Unit 2. All of these links go to the new folder. So if you're having trouble accessing the folder, you can get it once you get to one document within the folder, that folder should show up for you, or you can kind of see where that navigates you to in your Google Drive. So we have a unit outline. The unit outline really gives a lot more details. It's basically some condensed lesson plans. It tells you about timing. It tells you about order of events. I'm just opening it here so that you can see an example. If you've been, and right now you can see my my comments on your end. When you open this document, you can't see the comments. Those are things that we work with, or the comments that we make in the Biology Council, which is the group that develops this curriculum. But you can see that we have, task set 1, and what the essential question is, the phenomenon, and then a variety of tasks. There's information in here about which practices, cross-cutting concepts, and core ideas are being covered, what kinds of assessment you might be able to do with that particular task. And then on some, you also have access to extension information, like what additional information could you give to students who may need more, right? Or may need something that they're doing on their own while others are doing something different in a class. So it's an opportunity for some differentiation. There are links for keys in here also where they're available. I will say, we have checked all these links, but it's possible whenever you're doing a big transition from Drive to Drive, we're finding that sometimes we are making mistakes with linking. So if you find that a link is broken or in the wrong place, if you could we do have a feedback form that I can tell you that is actually linked in the bottom of our slides for today. And that feedback form will help us resolve some of those issues. I've also linked for you in our slides on slide number 10 for today, 2 different versions of a interactive notebook. I personally use the Google Docs version of the interactive notebook in my class, but we have colleagues at Sunset High School, and my colleague, some of you know Jesse Chamberlain, who worked on the initial version of this slides, but our colleagues at Sunset really have advantaged it. Okay, in the presentation, it looks like the genomics interactive notebook. Okay, good. So thank you. Already my first link mistake. You can certainly get to the interactive notebook from the Biomolecules thing. Let's see if I can figure that out. I'm going to change that, copy that link. Docs. Oops, that's not going to work very well. I'm going to have to fix that later, but thank you. Or maybe Scott, you can fix it in the background. That would be helpful. Thank you. So I already kind of gave you a little bit of a preview about our engagement activity, what we like to call the unit opener. So there's a scenario about baby Matthew that I shared with you earlier. It's adapted from the National Center for Case Study Teaching and Science, which used to be at the University of Buffalo, but NSTA has now taken over kind of the ownership or the management of that group of case studies. So I think that if you're an NSTA member, you can get access to the keys for free, if the case studies themselves, if you're not familiar with them, I think that the materials themselves are free, but the keys you have to be a member of NSTA to access. So we present the scenario about baby Matthew. Students read a text, and then they ask questions using Jamboard like we did earlier, or they can also use sticky notes. So if you prefer to do some materials or some things non-digitally, you can certainly do a driving question board using sticky notes. And there are notes in the slides to help you facilitate either version. And driving question board is really kind of a hallmark of patterns science courses. The goal is to try and get students to think about an open-ended questions that could be answered by the unit that we're trying to or that we're covering. So when we do driving questions boards, it's true that sometimes students are asking really great open-ended questions that aren't actually in the scope of the unit, but it gives you an opportunity as a teacher to have some of those side conversations sometimes and trying to be able to answer the driving questions that students are asking that might not be a part of your lesson, or your lesson sequence. So I really like to do the driving question board. I keep those up if we do them on stickies, I do them on poster paper in the classroom, and I have one for each class, and we're able to group them and to try to get those answers to as many of those driving questions as possible. Inside the interactive notebook, there is something called a learning tracker. At first, I didn't really use the learning tracker very much, but now I use it pretty consistently. I ask students to do some kind of reflection about their learning at the end of every task. And the goal of that is for students to have kind of a running commentary about what they've learned. And when they get to an assessment, they don't have to go back and think to themselves, well, what did we do there? What did we do in this task? They already have a identified reflection that they did while they were in the moment, just after having done the task. So I find that that's very helpful for reflection and review. So after we introduce the scenario, and that usually takes it's only about a half a block. I'm on a block schedule that takes about 45 minutes, so half a block. Half of one class period for me. We jump right into what are biomolecules. And if you've taught any other patterns courses, patterns physics or patterns chemistry, then you are familiar with the idea of an inquiry cube. Now, in those courses, the inquiry cube is taught like the very first day of class. And in biology, we don't introduce that on the very first day of class in the fall, because the first unit is about ecology and it's not about biomolecules. And our inquiry cube that we have for this course has been devised around monomer-polymer pairs. And so I have a picture of what the flattened cube looks like. And if you're not familiar with an inquiry cube, the basic idea is that students are working in their table groups to identify patterns that appear on the cube, but they're not allowed to touch the cube. They're not allowed to pick it up. And they're trying to figure out what is on the bottom of the cube. And so there are a series of patterns that they can find. They're all surrounded or around the idea that opposite sides of the cube are monomer and polymer pairs. And they're also color coded. So you as biology teachers can, I'm sure, all figure out what's on the bottom of the cube. Certainly if you have done this activity before, you know what's on the bottom of the cube. But this is not necessarily a teaching like a memorization task, right? For students to memorize what the biomolecules are, but for them to practice collecting information from a device that is also presenting them with vocabulary that they may or may not know. They probably know the word carbohydrate. They probably know the word protein. I have noticed that in the last 5 years, more and more students know the word amino acid because they all have gone to GNC and have bought protein powder. So they don't, however, know the word nucleotide, or many of them don't know yet that word. And so they have, even though they probably have heard of the term that's on the bottom of the cube, which by the way is DNA, they've probably heard that term before, but they sometimes have a hard time getting to that or figuring that out because they don't know the word nucleotide. So there is we updated this document, or we updated this task during CDL. So there's now a video of the inquiry cube, so students can totally do this. If they need to be at home, there's a video of me turning the inquiry cube around on my desk. And there are images of all of the components of the inquiry cube that are embedded into the slides, so they can see those very easily, without, even if they don't have a physical inquiry cube in front of them. We follow that up with a task that's a card sort about different kinds of molecules. And this is pattern matching, has been kind of circulating around on the internet for a really long time. I've tried to find the original the name of the person who originally wrote it, and have never been able to find it, but I've really tried very hard to find it to make sure to give that person credit. Can't figure out who made the original version of this, but I'll just bring up the Jamboard so that you can see. We ask students to take a look at all different kinds of molecules and we give them their names, and we ask them to look at the characteristics of these molecules and to classify them in groups. Now, on the teacher side, I know that I'm trying to have them group these by similar shape, similar R groups, or similar types of patterns within the molecule to get the groups of biomolecules that we will be talking about in the rest of the unit. But they don't necessarily know that at this point. So this is really an investigative task where they're able to see how the bonds that they learned about in chemistry work to create different kinds of molecules that are used in living things. There are two versions of actually three ways that you can do this. You can do it with the Jamboard. There are two versions of the Jamboard. One of them has lots and lots of molecules. The one I just showed you was the one that has all of the molecules in it, including steroids and some long chain fatty acids. There is a second version that's a little bit less cumbersome. It has fewer molecules. It only deals with the main molecule groups. If you want to do something that'll be a little bit less word, I mean, it's not wordy, but too many molecules sometimes is too much. So you can do it with the fewer molecules, or you can do it with a card sort. So I forgot to put the link here. Maybe Scott, you can add that link in to the paper version. You can print out the cards on cardstock and cut them out and have students physically move the cards around on their table and then take pictures of their groupings. So the goal of this task is really to have students notice that different groupings of molecules have similar characteristics, and that allows them to react chemically in similar ways. We don't get into the fact the differentiation between different R groups and amino acids, for example, just recognizing that the R group exists, and that because amino acids will come back a little bit later in the unit, and we can come and readdress that question at that time. Good. Scott says that the paper card sort works really well if you are not one-to-one with computers in your classroom. Right. And that's, of course, what we did before CDL. The Jamboard components were created when the Jamboard versions were created when we went to CDL. All right. So once they have a general idea, students have a general idea about the different categories of biomolecules and the different functions that those molecules have and the structures of those molecules, we can get into a little bit more details. Valerie says, oh, would you modify the card sort if students haven't had chemistry yet? So depending on, I think that you can probably still do it if they haven't had a chemistry course, because they've probably had some access to information perhaps in middle school about what a chemical bond is, as long as it's, maybe or maybe not. So good. Holly says that she does it without fully understanding necessarily what they're looking at. They can still figure out the patterns that we're trying to get them to see. Thanks, Holly, for that. And Carissa, thanks. So Valerie, looks like yes. I mean, you'll have to look back, you look and make whatever, modifications that are necessary for your classroom. But the goal of that is not necessarily to know about bond energy or anything like that. I think the only thing I haven't taught this yet this year, so I'm like digging in my mind for what kind of chemistry knowledge would be really necessary. One, I would say, is just recognizing that carbon is an extremely versatile atom, right? That it has 4 electrons that can bond, and therefore it can make four different bonds. They don't really have to know that, but it definitely helps when they're looking at the molecules, looking at all of the different kinds of molecules that carbon can make because of its versatility in bonding. But that information is easily shared verbally. Yeah, patterns matching poster is an option, right? To sort the cards. And then there is good. knowing skeleton structures, like I put a link to a video. I think there's a link to a video in the slides that talks about skeletal structures, like the idea of, the carbon atoms not always being present in a diagram because it just gets too cluttered. So there is that's not even something that my students get in chemistry necessarily, especially not during CDL when we had to kind of pick and choose most important information. So there's a link in the slides to a video that describes that. So once they have a general idea about kind of what molecules we're talking about, we have a couple of options for them to dig a little bit deeper. So there are a couple of options here, and I will say that options are one of the things that has really become something that we've been working a lot more on in patterns biology over the last, I would say, year to year and a half. During CDL, certainly, and then as we've gone back into the classroom, we're trying to build options. Sometimes the options are going to be for a level change, like if you might be using patterns materials at a level different than 11th grade, but it also might be an option that takes less time or that is less detailed. All of the options still address the performance expectation and are rigorous, but some of them just inherently take less time. And so because they're approaching the topic in a little bit of a different way. So here's our first option or set of options that we are going to be talking about today. The first one is a hands-on task that requires molecular modeling kits. This one, or a version of this, has been in patterns biology for quite some time. The students actually work with the molecular modeling kits to build simple sugars and then bind those simple sugars together, like a pair builds one and then another pair builds another one, and then they model dehydration synthesis to attach them together to make a disaccharide, and then they do hydrolysis to break those back apart, so to simulate those two chemical processes. And then they also do a similar thing with amino acids. Good, you can certainly use mini marshmallows, white and colored, to do this. If you don't have access to molecular modeling kits, that totally works. Anything that you can use to simulate atoms and bonds will work. And since we're basically dealing with carbon-based molecules here, you don't need tons of, I think the colored marshmallows, I think there are 4 or 5 colors, and that's more than enough. The second option, and I will say that hands-on one does take a little bit more time, and it takes some management because students have little pieces of things that they see as toys in their hands or food potentially. And so that does take some management. So for whatever reason, that's not the choice that you choose. You have another option, which is a digital option. It's a little bit less detailed. It's more generalized. And we pulled this document called Building and Breaking Down of Organic Biomolecules from something that was shared in the NGSS Biology Teachers Facebook group. Rachel Taylor is a teacher at Millennium High School in New York, and she shared this document or this task with that group on Facebook. And the great thing about her materials is that she does all of her own images, and then she often has videos on YouTube that match directly using the same images as she created for the documents. So I'll just show you what that looks like in this case. So all of these diagrams are diagrams that she made, and her diagrams are also present in her videos. And so we're getting back here to the idea of what's the polymer, what's the monomer, same kind of building on what we learned in the inquiry cube. So you can see that this is not the same as building molecules, but still gets at the performance expectation to know that carbon-based molecules build to create different kinds of, carbon atoms, and other atoms can be used to build different kinds of molecules and a huge variety of them. So those two options there in 2.3. You may have noticed that also we've kind of shifted away from using the term task set or TS. That was a decision that the council made. We got a fair bit of feedback that was complicated. And so we just kind of decided to go with a pretty standard unit number dot task number. just so that the recommended order is relatively clear. If you do the digital less detailed version, will it be sufficient to prepare them for the final assessment? Yes. And although you may need to, modify, right? The assessments that are in the folder right now, I think right now are only the assessments that I have given. One of the things that we're in the transition to the new folders is that we're archiving several old assessments. I think that what we're going to have in the restricted folder is a folder that's called archived or past assessments, so that people can kind of look through and get different ideas for different assessment opportunities, because that's hard to write a test from scratch on your own. So just be aware that those things will be populating into that restricted folder, but we want to only have, a couple of things. One of the reasons for moving to that restricted folder, or excuse me, to the new folders, is that those folders were getting overwhelmed and not organized at all. And even those of us who are intimately involved in the process were having real trouble navigating those documents. And so we're trying in these new folders, since we're building them from scratch, to have things be really clear and organized, but to also make sure that teachers have access to a variety of, specifically for assessment, a variety of options. Yeah, doing a review helpful. Yeah, and there are reviews also that we have written that are in too. Go ahead, question, Susan.
Yes, thank you. So if we had already subscribed to the biology pathways key folder before, is that now outdated and we have to subscribe to a new one?
Yeah, so you don't have to subscribe. You were already actually added to this folder. So it's in the shared drive. So if you navigate to drive.google.com with your school account, on the left-hand side, you should see, I'm actually going to open this up so I can demonstrate for you.
Thank you.
Oops, sorry, got to spell it properly. That's not going to work. There we go. On the left-hand side, relatively recently, like about a year and a half ago, shared drives became an option. And if you click on that shared drives icon, you should see, Susan, Patterns Biology, 2 folders for that already, because you were already subscribed with the previous. We added everybody to these folders in the summertime, and we informed people at that time, but we recognize that a lot of people might not have been reading their email, might have gotten buried in their summer email.
Oh, okay. And then can you just quickly say again what you had said? So the restricted one is the one that has all the keys.
Correct. So we're teachers just like all of you, and this is work that we do outside of our teaching day. So when we have keys that we have made, we put them in the restricted folder. So that would be like assessments. And so I'm just opening this up, for example. So here right now, there are no keys because I haven't moved them over. But those will be, as I give these tests in my own class, I'll be writing a key and moving it. But in the here, for example, for inquiry, we have an example of the conclusion for biodiversity in Unit 1. And then here is this example, a student example, right? You should automatically have access to all of that. If you're having trouble, we can troubleshoot that. We can troubleshoot that separately.
Thank you. I appreciate it.
All right. So speaking of assessment, here is kind of a first opportunity for an assessment. After they've done the modeling and learned about biomolecules, there's a quiz. I think it's a front and back if you're doing it on paper. You could use individual questions for students who haven't shown proficiency or are, struggling with some of the formative assessments that have come a little bit earlier. There's been a push since CDL to do some alternative assessments rather than just just tests. And so in CDL, we developed, or my team at school developed an infographic option that connected to nutrition and all of the reasons that biomolecules would be needed in nutrition. And it has students track their nutrition intake. We had some students that did really amazing work with this, and it worked as an alternative assessment for us in CDL. But it's certainly something that's open, available for you to use. And then, of course, at the end, they're doing an ongoing formative assessment in their learning tracker. That's in their, for me, in their interactive notebook. My teaching partner this year is using a physical notebook, and she's having all of the students write in the physical notebook, and she's doing physical notebook checks. I do it by Google Drive, and I just every couple of weeks or so, I'll open up all of their interactive notebooks and give them a formative score based on what they've been able to demonstrate. So of course, if chemical reactions are happening in cells to build and break biomolecules, we know that all of those things are facilitated by enzymes. And even though enzyme action is not called out specifically in a performance expectation by the NGSS, the patterns biology team has always felt that including enzymes in this unit is an essential component. Not to mention that it also is really important for understanding what's happening with our phenomenon with baby Matthew. So we do have an enzyme lab, and those of you who have taken the in-person summer class have actually done this lab in person. And we do also have options for virtual for students. And those of you who have taken the summer class online experienced this. But certainly before doing a lab about enzymes, we have to understand a little bit about enzyme function in and of itself, because the lab focuses more on limits to enzyme function. So there are a couple of new things in this in the past couple of years. There's some, although we don't take notes per se very often in patterns biology, this is one of the times where we do that. So or we ask students to do that. So there is a note taker. The note taker is embedded in the interactive notebook if you're using that. And that goes basically to the level of like lock and key hypothesis. At regular biology level, we don't do induced fit hypothesis. Especially this is just something that, because it's not called out specifically in the NGSS, we want to make sure that they have enough information about enzyme function to be able to be successful at understanding how enzymes generally work and how they can apply that to the idea of the baby Matthew scenario. But they don't need all of those details about induced fit hypothesis. After they get some general information about enzymes, this is a new task. They do or have an opportunity to do a investigation about different kinds of metabolic enzymes. And this is something that I wrote after students just kept asking, like, well, what are some other enzymes? Like, because there are some examples of enzymes in the, kind of the notes section, but they really wanted to know more. And so I wrote this to fulfill that purpose. So we use catalase as an example, because of course that's what we'll use in the actual lab that we'll be doing in the next bit. And you'll see that there's a list of different kinds of enzymes, and there are about 20 here, 22, 23, because many of us have class sizes that sometimes range up to 40. So if you have 2 students working on each enzyme, you have plenty of enzymes here for each pair to have a different one. And the goal is to try and figure out, well, does this enzyme build biomolecules or does it break biomolecules down? Is it a builder? I like to say, is it a builder or is it a breaker? And then I asked them to find out what a picture of that enzyme is, so they can see a model. And almost all enzymes now, because of the modeling that's available, computer modeling that's available now, if you have the amino acid sequence of an enzyme, you can generate what the physical structure of that would look like. And so you see an example here of catalase. And then the last thing we ask them to do is to find out what the function of that thing is, right? And so here are some terms that they can use, building, breaking, digesting, synthesizing, simple sugar, amino acid, monomer, polymer, all these terms that we've already addressed in class that they can use to help them write their explanation. So you can see, what's happening with catalase in my example, some interesting facts. And then I do ask them to reference, because it's important, I think, at junior level that students get into the habit of, first of all, recognizing that in science we use APA citations. They're probably familiar with MLA in their social studies and English language arts classes, but you talking about the idea that scientists also cite their sources and that they use APA format. So I ask them to put references in the slide notes. So it also gives them an idea about all the different kinds of enzymes that might exist. Obviously, this is a tiny fraction, but we wanted to use ones that were recognizable for students for the building and breaking of biomolecules in particular. What if your students have very little experience? So in the slide notes of this, I think I have a link to a couple of different citing sources online. The one, my current favorite right now is called MyBib, MYBIB. And I like it because it is a Google add-on or Google plugin. And you can also go to, just bring it up, mybib.com. The Google plugin basically does the same thing here, but I like it because it you can save your citations in a project. But if you use the MyBib add-on in Chrome, I don't know if you can see it right here, but there's a little, the icon that has a little arrow on it. And so let's say I wanted to go to, I don't know, okay, PubMed, let's say. So we're going to go to PubMed. We're going to find an article. I'm just going to put in my name. We'll see if my things come up. Nope. Okay, but we'll just pick up this one as an example. And let's say that I want students to cite this source. Well, first, this one happens to have a cite button right on it, so I could use that. And there's the citation. And actually, you can even change the format to APA. So that's great if you're using something like PubMed, where they're expecting that you're going there to access materials that need to be cited. But let's say you were even going to, like, I don't know, not Facebook, CNN. And you just click on the little icon up here and the citation generator pops up automatically and it creates the citation. Now, this is not perfect. I'm from the generation of people where I actually had the APA style book with a spiral. And some of you are nodding your heads like, you recognize, and we were, being graded in part on whether we put the period in the right place or the parenthesis in the right place. That is not my focus with this. I recognize that MyBib is not perfect, but my goal in this time at this age for students is to get them into the habit of citing their sources. So if they make their best effort to cite the source and it looks like this rather than just a URL, then I give them, quote unquote credit for that. Right. So and it also shows the in-text citation. And notice here that if I click Save to Project, it will actually save that in my first project. And I need to change this now to APA. But it automatically changes things from APA to MLA or whatever version you want. If you're using Harvard or Chicago or whatever style of citations you're, the people that you're writing for are wanting. But generally speaking, in science, APA is what is used. And so that is what I try to model for my students. So you'll start to see also in more and more of the patterns biology materials that images are cited now, because that's really important to me to model that behavior, that if I'm getting something from another source, that I need to be able to cite where I got it from. That helps them see that even adults have to do that. Right? This is not just about them. Okay, so yeah, so we're looking at this. So this is one of these, this is an example. Whenever it says template in the title, you as a teacher would need to make copies of this slideshow, one for each of your classes. Yeah, I know. Seriously, Valerie, right? Like that would have saved me so much time. But then again, I'm also from a typewriter generation. So we had my very first computer halfway through my college career. So really, things are very, very different today. All right. Other questions about any other questions about citations or this metabolic enzymes task? I just added this in, or worked to create this because students were asking about other examples, and I found that only by giving them a very restricted number of examples, they didn't get the idea of the scope of the metabolism. Right. And so all of these enzymes that they're investigating in this are metabolic enzymes that function in their cells, in many cells, not all cells, but certainly in human cells. So then there is an assessment opportunity here right now. This one's built in Formative, goformative.com. So you may or may not have access to build your own formatives, but one thing that's great about Formative is that you can, with the website formative.com, is that you can use, if you click on this link, it will recreate that for you in your Formative files. You can certainly also use those. That would be easily convertible to a Google form if you wanted to do that. There's also another opportunity to do something hands-on here. Some of you may be familiar with toothpick case, which is a classic activity in AP biology that talks about enzyme function and limits on enzyme function. Back in the day, we used to do this as an introduction before doing the enzyme lab. I didn't like the idea that toothpicks were just going to get broken and thrown away. So I created a version of this for pop beads so that the pop beads can be used over and over again and not have just materials being trashed. That's an optional one. Sometimes I do it if I have an extra day, and sometimes I leave it out if I'm short for time. And then once they have some general information about, or knowledge about how enzymes function and what they're, what they generally do, is we jump into the enzyme function patterns inquiry. And just going to put a plug in. I think it's on Wednesday. On Wednesday, Wednesday, maybe not. I can't remember what day it is. Somebody will be able to tell me, Kristen or Bradford, somebody will be able to tell me what day it's on, where PMSP is offering a Desmos webinar. So if you are feeling like Desmos is not a strength for you, then I would really encourage you to go to that webinar and get paid to go to do some professional learning around Desmos. The patterns biology team really tries to make the Desmos process as painless as possible, but that doesn't mean it's without difficulty sometimes. I remember me learning Desmos. I had never used Desmos before about 5 years ago, and it definitely had an impact on I had to spend a lot of time figuring it out. I would have loved to have had access to a Desmos webinar to help teach me. But if you really want to be successful in helping your students understand the limits to enzyme activity, it's really important for you to know how to use Desmos, because this lab has some of the more complex mathematical patterns in it. And if you don't know how to graph a standard distribution in Desmos, then you will not be able to help facilitate your students doing that. So I would really put in, and Bradford, are you facilitating that?
Yes, I am.
Okay. So I may just send you a standard distribution example that you may just be able to throw in as an advanced highlight. Because you may sounds like you're going to have a fair number of biology attendees who might need to know how to do that, but I can get you that.
Sounds great. Thank you.
Yeah. So some updates on the patterns enzyme function lab. In terms of the doing of the lab in the classroom, no updates really. The patterns are the same. It's a jigsawed organization in that there are 4 different things that student groups can test. They test how temperature affects enzyme function or enzyme activity, how pH affects enzyme activity. Although I'm going to put out a call for all of you. If anybody has really good tips about how to get the pH one to work consistently well, I'd really love to hear those, because it seems like every year I get differing results with that, whereas all the others are pretty standard. So if you have tips about how pH has worked in your classroom, I'd love to hear them. The third one is enzyme concentration, and the last one is substrate concentration. So these 4 typical things that students learn about how to generate, or how enzyme function is affected by those 4 things. It's a jigsaw. So in my classroom, sometimes I have up to 10 groups. So I have, on average, 2 or 3 groups testing each variable. We generate all of those patterns. Each group generates their own pattern. They do their own investigation. They do their own data collection. They do their own graphing. And then we engage in a data analysis across groups. And there are 2 options for you how to do that. One would be and this is the one I use a data sharing slideshow, where the students are given some instructions about what they need to include on their slide. And then I have all the different groups represented here, and you can copy the slides down here as many times as you need them when you're making your own copies. And then what I do in my classroom is that I actually have students break into small table groups. So at each table group, I have one person who studied temperature, one person who studied enzyme concentration, substrate concentration, and pH. And they become experts on their own answers first, and then they present those to the people at their new table. So yeah, so good, Valerie, questions about that. We did make some suggestions in the teacher notes about that this year. So there's some updated teacher notes that you might want to take a look at. Yeah, temperature variable is definitely the more complicated one. Holly says that she used hand warmers, insulated mugs. I have a sous vide device that I use for making yogurt that I bring in. It's much, much cheaper than buying a water bath. But this year we were relatively successful at doing using a cold pack and putting like a cold pack that you might put in the freezer to keep things like a sandwich box cool. Sometimes they come in like those insulated lunch bags. You could put a ice pack in there and put a beaker of water on top of the ice pack before class starts, and that cools the water down just enough so you can have like an ice bath, a true ice bath, and then something that's a little bit cooler than room temperature, maybe like 10 or 12 degrees C. Then you have room temperature. Those are all relatively easy to control. And really, then the only one you really need is like one hot one if you can get it. Sounds like Holly was able to do that with hand warmers and insulated mugs. Because I think we use potatoes for this catalase extraction. And they tend to denate, those enzymes tend to denature at like 40 or 45 degrees C. So it doesn't need to be boiling hot. Oh, great, Erica. Yeah. So the sous vides are so much easier. Like you can get a sous vide device, I think, for like $60 now. And a fancy water bath is like almost $1,000. So it's a huge, huge difference in price. I usually bring mine from home. We do have one in the department. And then we have another science teacher who brings hers from home. So I have access to 3, but we have to run 2 classes at the same time. So we have to split them. But you really only need one at the warmest temperature. Another option for doing the data sharing is to do a data discussion notes document. I know that they're doing this We developed these data discussion notes. I think that in physics and in chemistry, they're doing a lot more of this now, this type of data discussion. This worked relatively well during CDL, but it's just another way to facilitate the data discussion process with some reminders. So depending on your preference of how to do the data discussion, you're welcome to do either one. There are some materials also in and they have been copied over to the new folders about different ways that you can do data discussion. Traditionally, the whole group method is kind of the easiest one to do, but data discussion facilitation is really an art. If you have a person in your building or who you're able to go and see or watch in some way, or, get in on Zoom sometime to watch somebody else facilitating a data discussion. It's not something that's easy. There's a relative amount of discomfort that the teacher undergoes when they're working through doing data discussions the first few times, or maybe even the whole first year or two. So don't feel discouraged about facilitating data discussions, because it is a difficult thing. But one of our patterns design principles is about facilitating student discourse. And it's really important to persevere as a teacher through those difficult moments of when your students are not talking in a data discussion. It's very easy to go back into the mode of I'm the teacher and I will just share with them what the patterns are rather than having them discuss. But really want to encourage you to use those data discussion techniques. And if you can, go and check out others who maybe have some more experience doing that, so that you can refine your practice. For the assessment, I collect the data and conclusion for this task as an assessment. I like it because it incorporates both content knowledge and practices. And so it is a nice way to hit multiple types of targets, depending on what your system is for learning targets, if you're assessing skills and content. And then after that is another thing that I built last year, or I guess during the CDL year, to reconnect the learning around enzymes to baby Matthew. And so I built, and Jesse Chamberlain helped me build this also. We wanted to demonstrate what a metabolic pathway looks like, because up until this point, we're talking about enzymes really individually, separate from each other, and not talking about how they interact or how they might work in a pathway. And so I built this pathway that actually shows how glucose, of course, a simple carbohydrate, can be converted through, and I think it's a 9-step pathway into an amino acid, glycine. And so I've color coded this. All the green ones are carbohydrates, and then the red ones are considered on the proteins, because they have nitrogen in them. And so what students do is that they see the pathway, and you'll notice that there are some blanks here, and these blanks get filled in. On the next panel, they have the 5 molecules that go in the blanks, and they have to analyze the structures of the molecules to try and figure out which molecule goes in which blank. We've given them the names of them, not because we're going to test them on the names, not because we expect them to break down all of the names, but to help them see patterns. For example, glucose 6-phosphate. If they read the name and they are able to connect phosphate to phosphorus, then they can pretty much figure out that they can find a glucose molecule, which, by the way, is this one, right? The 6-sided one, and that it has this thing with phosphorus sticking off the corner. They often want to pick this one, which is a 5-sided sugar. It's a pentose instead of a hexose. Go ahead, Susan.
So I'm wondering, how much of an example, like how far should I go with giving an example, giving them information? And how much should I leave it to them to like kind of struggle with?
I give them nothing. I give them this, and then I walk around. And I say, so the very common mistake that they make right away is to put this pentose first, and they don't pay attention to the shape of the sugar. So I am walking around during this whole time and I'm pointing out, hey, does this thing, like they might take this guy, I'll just show you an example. Ooh, what am I doing there? I am erasing. That is not what I wanted to do. Backwards. Definitely don't want to erase. So I'm just going to cut this out and I'm going to paste it here. This is exactly what students would do. They would just paste that picture and then drag it to the right spot. So if I see them do that, I would say something like, hey, does it make sense that this thing would, because if they do that, well, sorry, one step more I have to do. It's very common for them to do this. And I ask, well, does it, what might be happening for this 6-sided molecule to turn into or to be converted into a 5-sided molecule and then be turned back into a 6-sided molecule? That doesn't make a lot of sense, right? So I'm kind of asking those open-ended questions, like, why did you make that decision? Did you look at the patterns in the shapes of the molecules, which by the way, is something that they did when they did pattern matching 3 or 4 lessons ago. So I'm able to refer them back to those ideas about looking at the shapes of the molecules and what's on the ends of the molecules to be able to make sure that they're placing things correctly. So I let them struggle. I tell them you're going to cut out the molecules on panel 2 and place them on panel 1. And then I say, you guys got to figure it out. Let me see if I get these items back here so I can.
Okay, and then
That's my
after walking around and then kind of coaching them through it, do you share like let's go over the answers at the end?
So good. So that's a good question. So what I also ask them to do, I ask them to circle, and my students we're lucky we have their Chromebooks have a stylus and a touch screen. And so what I ask students to do, and I think I have some there are examples in the slides that address this or that show this. I ask them to draw a circle around the parts of the molecules that are changing with each reaction. So for example, glucose 6-phosphate is adding, or the phosphate group is being added to glucose. And for those higher flyers, that helps them realize that a kinase is an enzyme that adds a phosphate group. And that actually shows up here too, kinase. And there's another kinase here. So, or a phosphatase would be cutting off that phosphate group. So I will say, Susan, that I haven't done this year yet. I would be willing, well, I probably give the answer if I have time in the class period. And if I don't, then I just leave it open. And I just make sure that they actually have the correct molecules in the correct space. But I wouldn't necessarily, if I was running out of time, I would just make sure that they had the correct molecules, that the pathway that they had developed was correct. But I would do that really by walking around rather than like presenting it at the end. They do also, if you do it this way, there's 16 panels in this Jamboard, so that each class only has 1 panel, but every group has their own area to work in. And so they have the ability, all the student groups have the ability to see what the other groups are doing, right? So that leaves it a little bit lower stakes, right? For them to be able to kind of check in on what other groups are doing. I don't really tell them that but they figure it out.
And then they can argue why they made those decisions as well.
Mm-hmm. Yep. And in terms of class time, how that would be spent, I would rather spend time on having students talk about the choices that they made rather than me talking and giving them an answer. So that's how I would, choose to spend my time. And then they can, like I said, they can look. There are invariably groups that are going to get it right, and the ones that don't get it right are going to be likely to go and check and make modifications. How do I share the Jamboard with the students? We use, I give a link to the Jamboard in their INB. So I have to prepare the Jamboards in advance because I'm using a Docs INB, which I can't edit after I've given it to them. So I prepare all of those Jamboards in advance and have them linked in their INB. But we also use Canvas. But if you're using Google Classroom, you could also just send it out to them via Google Classroom. So I have one of these per class. And I think there are enough in here for 8 or 9 groups, but it's easy for you to make your copy of this and then, adjust it to the number of groups that you need.
Later, can you share like a sample of your INB so that we can kind of gauge ideas?
So there's a couple of ways that you can find it, Susan. So if you have these slides open, I think it's on slide number 2 or 3 or early on, and I have some resources for Unit 2, but you can also get access to the INB by clicking on the outline, and it's linked in the upper right-hand corner.
Oh, okay, great.
So I felt that this glucose to glycine Jamboard takes their information, what they've learned about that every enzyme has a specific job, and those enzymes then can work in consort with each other to get a whole pathway that can go from one type of biomolecule to another type of biomolecule. Now, one thing I haven't mentioned yet is that in my classroom, I have a poster of metabolic pathways, and it's linked in this. It's in the Patterns Biology materials, and I have printed that poster out. It's a color poster, but it's also linked in slides, I think, I'm pretty sure, for these tasks. And I'm referring during all of these lessons out to that metabolic pathways poster, showing students how complicated the metabolic pathways in cells are. But that really there's kind of like a pathway from every kind of biomolecule to every other kind of biomolecule if you have all of the enzymes that are necessary, right? So that was a piece that I felt was really missing on the student activity end. And that's why I developed this glucose to glycine Jamboard with Jessie to really help hammer that home, that enzymes have to work in order. They have to have these pathways, or you don't end up with the products that you need. And that helps us answer our big essential question, which is how do cells build and break the molecules they need, and what happens if they don't have those. Okay, so this is a change in position. So we used to have this task at the very, very end of the unit, but it seemed extremely disjointed. So since we've just finished talking about enzymes, we actually have moved or have suggested to move the yogurt engineering project to the middle of the unit rather than the end. So not a lot of changes in the content of this project necessarily from previous years, but in this project, students are actually engineering their own yogurt. They get to make some decisions about what to put in the yogurt, they do some calorie counting work, figuring out how much protein, how much carbohydrates, how much fat is in their yogurt, and then they analyze it. So this was a little bit tricky during COVID. So I know that, and Erica, maybe you can tell us, there's a team in North Bend that several years ago had converted this or applied this idea to making sourdough bread instead of doing yogurt. We don't have the capacity in our building to be baking 40 or 50 loaves of bread. So it's a little bit easier for us with our sous vide devices to do it's about a 1 cup yogurt sample. Erica, maybe you can tell us if there's been any updates to that. You can just tell us in the chat. We did use the bread project in CDL. A lot of students did that at home rather than yogurt. But in the classroom, in my school anyway, we've gone back to just doing yogurt. If we find out that the bread project has had some updates, we can link that back in as well. The students take on different roles in this project. Now, if you are a teacher who also teaches engineering, or maybe if you're teaching this on the trimester or have a little bit more time for engineering, There's an extension available to actually build the incubators in which the yogurt is being incubated, like to actually build the incubators and do temperature control. So if you're interested in that extension, I have physical materials I can give you, because I'm not doing that in my classroom anymore. I have lots of physical materials I could get to you that would help make that not as expensive, but also a lot of digital materials too, if you're interested in that. I will say that this is, when I run into students, this is what they remember about my class, is the fact that we made yogurt together, and some of them like it and some of them don't. And that's fine. It's a really great experience. And some students have never even thought about the fact that yogurt could be made at home or in the classroom. So.
I have a logistical question about the yogurt.
Yeah.
So when you were having all of your students make yogurt, I'm a novice at making yogurt. Can you put it in the refrigerator for all of your classes and then throw them all in the incubator at the same time? Are you dragging all of these home? Like, how does this happen?
So good. Thanks, Amy, for your question. So I put the teacher notes here on purpose, because this is if you have never done this with students before, there is a fair bit of context that's helpful from that we have tried to write down as much as we can. So I'll answer your specific questions first, and then I'll open up the teacher notes to show you. I do bring them home. I teach almost exclusively biology, so I probably have more yogurt samples than any other teacher. And sometimes I end up just doing it for everybody just because it's easier. Everybody has different, needs at home. I usually try to block out my calendar in the evenings on the days that I'm doing this, but I do bring them home. I use my sous vide device. Sometimes I bring the sous vide device home from school and do 2 coolers, depending on how many I have to organize. I also, you asked a question about like refrigerating them. At the end of my first period, I put their yogurts into the refrigerator, and they stay there until I take them home. And then I organize them by incubation time. And then I incubate different class periods yogurts together, as long as they have the same incubation time. So let me just bring up the teacher notes for you. You can see, like, there's definitely a lot here. We tried to organize it into, 2 days before. What do you have to do? Well, you've got to know, to find out how much milk to buy, you need to know what students' intentions are about what they're going to be, what recipe ingredients they need. Now, we restrict the recipe ingredients to make it as easy as possible. You can buy some of the materials in advance, like sweetened condensed milk. You can get from Amazon, and I just have several cans of that sitting in the stock room so that I don't have to go and buy that every year or every season. But you do have to buy a fresh yogurt culture, and you do have to buy milk. So we generally get reimbursed by petty cash if we can do that if you can do that in your building, or that's an expense that we take on and deduct from our taxes, as we all tend to do sometimes. Dry milk is another one that we buy online and then tend to have at home, or have in the stock room. Those things all, of course, have to be marked, food, and we have to have food use only, glassware, thermometers, those kinds of things. Because you certainly don't want students using glassware that has been used for other tasks that are not, food grade or food safe. If your chemistry team is doing the candy project, you could certainly use the same glassware for candy and for yogurt. They don't usually happen or at the same time of year if your teachers are teaching the patterns curriculum in order. Yes, I don't buy, it's bacteria actually, Valerie. So I don't buy bacterial culture in a lyophilized packet. I buy yogurt with live and active cultures. Yes. Yeah. Yeah. So there are, and there are certain yogurts that work and tend to be better flavor for students. Like they really, like I've had situations where every student yogurt that has been made has gotten ones on the taste test results because it's too tart. And so I tend to purchase Greek yogurt as my live and active culture. I tried Annie's and it was actually one of the ones that did not work well at all because it's so tart. So I think that the combo of bacteria that are in there were just really not aligned with the teenage palate. Oh, Annie's, not true. I've never used Annie's. I've used Nancy's. I'm just looking at my notes here. Nancy's is very acidic and sharp tasting, and that has not worked. But Annie's, I'm not familiar with. Is it the same ones that make like the cheddar bunnies? I don't know that brand. So I generally use, okay, yeah, Nancy's is very, very acidic, and we've not had good results on that. I don't mind it, but the students don't care for it as much as the ones that are milder. It's all in the teacher notes. I've used Faye and Zoe unsweetened, but you can also use the store brand too. But all of that information is in all of that trial and error that we've done is listed in these teacher notes. We have suggestions about what to do on the day of, how to set up your classroom, a goal. One thing that we'd like to try and do this year is to add pictures to this so that you can kind of get an idea about how we've set things up in the classroom successfully. Some areas where you can, like things you need to keep an eye out for students who are, if they're heating up their milk too quickly and they burn it, like that is definitely makes for not good tasting yogurt if they burn the milk. They also need to make sure to cool it down because the bacteria will die if they go into milk that's too hot. And then here, Amy, this gets to your question. I have some information in here about what I do when I bring all of the samples home with a timeline so that you can adjust your timeline based on, I get up relatively early. So I set them so that when I get up, it's the first thing I do. And then I bring the samples to school. We're on an alternating day schedule at my school. So when I bring the samples back to school, they are incubated in the refrigerator for another whole 24 hours before students get back to them in class. So we tried to provide as many of those things as possible. So those, you can see like this was last updated in 2019, these teacher notes. I have that in the bottom. But we are trying to do, that's another goal of the Biology Council, is to provide those teacher notes because that's a lot of institutional knowledge that for some of us who've been doing this for a while is all in our heads, but some of you who are newer to patterns need that information to be able to successfully implement. We don't want, lack of transfer of information to be a reason that you skip over a task or not do a task in your classroom.
Thank you.
You're welcome. All right. So any other questions about yogurt? This unit is kind of built into two sections, right? Or two big, like mini sections. So we have the general about biomolecules and a lot of focus on sugars and proteins in the form of enzymes. And then the second part of the unit, which we're just about to look at now, is the part about DNA and protein synthesis, how those proteins actually get made. So this is the part that if you're newer to patterns might be a little bit surprising that we're doing these standards, or the standard about DNA to protein at this point in the year. But you'll see how it does connect to our phenomenon. So depending on how you wish to approach, and this is another situation where you have some options based on what's going to work best in your classroom, to either do these two sections, 2.6 and 2.7 together, or to separate them, depending on level of detail and availability of materials and that kind of thing. So regardless of which set of options you select, I really like to do HKW in the beginning of this section. And this is like a modified, what we used to call a KWL. In my own experience, I would often do the K and the W on the first day, and then we would leave the L for later, and then we would never, ever come back to the L. So I prefer to use this modified version, which is an HKW, which is what have you heard? What do you know? And what do you want to know? Because a lot of times students assume that what they've heard is something that they know, and that knowledge is actually accurate. So what they've heard, I try to differentiate that from what we know to be true. Sometimes there's certainly overlap there, but sometimes there's not overlap, or sometimes they think that what they've heard is true and it's not necessarily true. So I like to use that modified HKW instead of, older style KWL. So they're, obviously talking about DNA at this point. And since this is not the first time they're hearing about DNA, because DNA was on the bottom of the inquiry cube that we did earlier on in the unit. And we have, up until this point in talking about enzymes, talked about the fact that DNA is responsible for determining, genes are responsible for proteins. So not in any detail at this point, but those questions certainly come up in the classroom, and I don't shy away from answering those questions. I just maybe don't go into super detail if they come up before this point. So there are a couple of ways that you can go about addressing these two sections. We do have a patterns inquiry here, which is written around Chargaff's rules, and students use a model to help them analyze the composition of DNA. So this is where they're figuring out what the components of a nucleotide are, how the nucleotides are put together, how the nucleotides attach to each other, and they, depending on your what you have available or what you want to do, you can either give them the model and have them analyze the model, or have them build the model based on some data that they analyze about Chargaff's rules. And if you go about it, in that way and use that DNA structure inquiry, then there's a separate task about protein synthesis. However, if you want to do something that's a little bit less detailed, and again, both of these options address the performance expectation, just at slightly differing levels of detail, but they do both fulfill the performance expectation. There's a less detailed alternative that combines the idea of DNA structure and the there's a typo there. Sorry, not protein and synthesis. DNA structure and protein synthesis in the same task. That still, you could also chunk that one into the DNA structure part and the protein synthesis part, depending on your timing in your classes. Some of you are on a trimester and do a 70-minute period. Some of you are on a 45-minute period. Some of you are like me doing a block schedule. So you have lots of flexibility depending on which of those tasks that you want to use in your classroom. I really do like to use the models. So students are in this one. This is still tries to use a similar type of strategies that we have in other in other inquiry tasks. But they're looking at the mixture of components in the drawing. So I'll show you what the drawing looks like. So so you can see that we have nucleotides here, and then each nucleotide has parts. And so they count how many P's, how many phosphates are there, how many S's, how many sugars are there, how many of these other little colored things. Those, of course, are the bases. How many of those are there? And then they use that information to help identify what the ratio is. And then they put them together to create a molecule of DNA that, let's see if I have that. I think on this one, I have the key of what that molecule looks like. Yeah, here it is. So here's the DNA, the kind of molecule using those same pieces that have been put together like a puzzle. So you have a choice whether you want them to do that putting together of the molecule, or whether you just want them to do the counting of the components from this pre-built model. You know, old style would have been, we would have just stood up at the front of the room and said, Shargaf was the scientist, and he calculated that A and T are equal and C and G are equal, right? That it's true that the same information is getting across, but the fact that students have agency in doing the counting and figuring that out for themselves aligns much better to our design principles and patterns. So once those things have been addressed, right, it cover, and I would say that those are probably the most complex ideas in this unit, right? Protein synthesis is a really tough one, but the NGSS performance expectation around protein synthesis is actually way, way, way less than what we used to teach. So don't feel that you have to get into the nitty gritty of that. A site, P site, like I'm not even sure I talk about transfer RNA really very much in protein synthesis in this course. If students are needing that, there are resources available to them, videos and the like that are embedded in the lessons where they can see that information, but I'm not testing them on transfer RNA or anticodons or anything like that. Because the key piece that the performance expectation is trying to address is that when there are mistakes, when there are mutations in DNA, that's going to have an effect on protein function. So this is really more the meat. We get them where they need to be to understand that genes connect to protein function. And here's the applicable skill, which is recognizing what happens when those proteins are not working correctly. So this muscular dystrophy task has been in the unit for quite some time, but it did get an update during CDL. So if you haven't looked at that in a while, you may want to look at that. There's certainly some slides for student support. And so this task, of course, talks about different kinds of muscular dystrophy. It also helps them compare and contrast between different mutation types and to understand that a deletion or an insertion is likely to be more problematic for enzyme function than a single substitution. And so they kind of compare and contrast between multiple different types of mutations and then are able to extrapolate about that to which type of muscular dystrophy might be caused by which type of mutation. And all of that is connected back to actual science, like the actual mutations and the actual conditions, versions of or types of muscular dystrophy. An optional follow-up, which kind of goes back to the idea like the metabolic enzymes that we looked at earlier, is another Jamboard where students have some options among which to choose. So we found like 40 different proteins. And I have them all on this Jamboard, and each student then picks a protein and edits the sticky note to tell us what happens when that protein doesn't function correctly. So for example, with lactase, it's an enzyme, so that's the kind of protein it is. And then when there's no lactase or when lactase isn't functioning correctly, your cells can't digest lactose. And so in human beings, that presents as digestive discomfort when you ingest lactose, right? So lactose intolerance, right? So that's the example that they've heard of before. And then each student researches one of these proteins, chooses one sticky note, and tells us about what happens when that protein when the gene has a mutation in it and resulting in a misfunction or malfunction of the protein. So it's, again, another way to kind of come back to the idea of students realizing that these are problems that could happen across the metabolism. There's another more like a written assessment opportunity. There's I call it a mini test that covers just the DNA to protein to trait, and it also covers mutations if you wanted to do that. And then they're filling out also their learning tracker. And then we just have one more. Oh, and I'm realizing, Scott, maybe you can fix this. I have the wrong unit outline present on this slide on 2.9. If you could fix that, I'd appreciate it. The last task in the unit is to come back to the baby Matthew scenario. And piece of feedback that we have received for, around the, during COVID and when people were really spending a lot of time, having time to reflect and provide feedback. One piece of feedback that we got over and over was, this unit seems really disjointed and we have not been doing we don't have regular check-ins about baby Matthew built into the unit. And so we really tried to do that with the thinking more about metabolism and making those check-ins and doing the pathways and that kind of thing. But this is really where we're doing our last check-in and kind of answering the baby Matthew scenario. So so what students have an opportunity to do here, and there's a couple of different ways you can do it. We have it set up informative, and then I prefer to do it on a slides template. That works well for me. There is a poster called Inborn Errors of the Metabolism, and I do have this also printed out in my classroom. Obviously in the electronic version, when I'm showing you here, you can't read all of the information on the sides, but each one in the margins here is the name of a metabolic condition that has been defined as as being caused by a deficiency in one or more enzymes in a pathway, in some kind of metabolic pathway. And so we have selected, I think, about 15 of these, and you can see in the slides template what the names of them are. So there are, 15 different ones, and student pairs work together to figure out what causes this condition, what enzyme is involved, whether it's a problem that's related to a buildup of a toxin or not enough of some kind of necessary molecule, which one of those scenarios causes the condition to occur. And then they write a little CER where they're making a claim and providing evidence and supporting all that with reasoning about whether they think that the condition that they have researched is the one that is affecting baby Matthew. And then we do a share out where each pair of students shares their information with the class. And actually we had a really great idea that came up in our summer class this year to build a bracket and have students to share at their table groups first and then kind of narrow it down. That would prevent the amount of time in the classroom where one person is talking and everybody else is listening and not necessarily writing or doing something. So that's on my radar for trying to figure out how to do that this year, how to facilitate that. But they're writing a claim evidence reasoning with their partner about whether they think that that condition is what's going on with baby Matthew. I did do during CDL, I made a video example of the process, because when students are doing this research, they do come across lots of, oh, great, Mary, that sounds exciting. Presenting their claim like a doctor at the front of the class. That's really cool. I did do a video example because when students are doing this research, they do come up against a lot of technical terminology. And so what I tried to present in the video example, that they need to persevere through some of those difficult words. They don't have to understand every word they're coming across in their research, but they do need to make sure that they're trying to pull out the essential pieces to be able to make a claim by the end. So Mary, I love that idea. There's a variety of ways that we can incorporate real life experience, and because that's what doctors do in rounds, I think, right? I'm not a doctor, but I think that the symptoms get presented, and then I think that's part of what students do when they're medical students, is to try and figure out what's going on. At least that's what they show on dramas on television with medical students. So I've covered the content of the unit, and I do want to set aside some time for questions and answers, but I just want to really quickly point out to you that I have a variety of things at the bottom of our slideshow that talk about, or that give you some information about discourse, because this is something that we know is difficult to do, right? Especially coming out of COVID, getting students to talk in your classroom is sometimes feels like it's almost impossible. And so here are a series of things that we have figured out over the course of time that work well to help get all students engaged in discourse. I've provided you with, here's some examples of norms that you might put in your classroom that help support discourse. Here, I think this one is from elementary, but you have some, accountability pieces that you can post. This is something that I have actually taped to every table in my classroom. I put the link there for you so that you can print them out also. I tape these to my tables so that at all times students have framing available for them to help them communicate more effectively with their table group or their shoulder partner. And then there's also a whole lot of research about talk moves, right? Things that you can say or that you can encourage students to say to help facilitate or move conversation forward. Some information here about differentiation. I'm just going to turn over to questions and answers. I addressed the summative assessment opportunities, but you have a list there that's also at the bottom of the unit planner or the unit outline. Other supports. If you don't have access to the shared drives for some reason, you can fill out this Google form, but that would really only be the case if you have never really done anything with patterns before. Most of you have been on the old folders, and if you were on the old folders, then you should have access to these. But if you're having trouble, we can help troubleshoot also. So we have about 7 minutes for question time. How do you close the one-on-one conversations? Do you share the right answer? That's a good question. So Jorge, when I do one-on-one conversations, so I like to say that in my classroom, raising of a hand is to ask a question, not to provide an answer. So I actually run my classroom mostly by cold calling. But obviously we have students who, for maybe it's on their IEP or their 504 plan, especially now students who suffer with anxiety. I do those one-on-one conversations kind of in advance of being called on. And so I might go to a student and say, hey, your turn's coming up. Here's the question I'd like to ask you. And I get that student to tell me to work through their answer first. And we might do that during like a work time or a discussion time while the rest of the class is talking. I go and talk to that student and say, hey, let's work through your answer. And then they can just repeat it. And that way they can even write down their answer in advance so that they can read their answer instead of just feeling like they're being, taken completely by surprise. So I help them work through the answer before they're being asked to share. Suggested timeline. So Zaldi, I don't have it off the top of my head, but in the unit outline, there is each task has a suggested time frame, and then I think that at the top it says says what the total is. Okay, thanks, Carol. Let's take a look at your questions in the parking lot. Need to go backwards here. Okay. Good. So let's see. Yeah, thank you for that thing about the pattern matching. I think I've done that before, or found that link also. So patterns biology resources on the website. So you got this answered. Okay, great. So I think that the answer is no, but the reality is that we are all of us who are working on Patterns Biology Council are all full-time teachers. So everything that we're doing on this is outside of school time. So we are trying to make sure that everybody has access to the resources using the drive. That's how I access things. I go to the unit outline and then access everything from the unit outline. but it is on the to-do list that there's supposed to be a new website coming, but that's being managed by PMSP. Oh, what's wrong with baby Matthew? Baby Matthew has maple syrup urine disease. Can I tell you the Desmos equation for the enzyme lab? Well, there are 4 different ones. And I don't know who posted that, but maybe you can hang out and I can show you my keys after. I think I have keys are linked in the teacher notes. Potato catalase recipe. I run 1 red potato through a juicer, and then I dilute it 1-to-1 with distilled water. But that's also in the note. That's in the notes of the enzyme lab. There also, if you don't have access to a juicer, you can use you can blend it with a certain amount of water, and then filter out the solids. Do pH buffers work to alternate pH? Okay, that's a good question. I've never tested it with pH buffers. That could be a way to get around the problems that I'm having. Something to test. I would say that to get a good standard, I mean, we want to see low and high pHs having low activity and some pH in the middle having, a higher activity. If you're doing it with students, you don't want to do more than like 5 or 6 tubes, because depends on how many test tubes your test tube racks hold. If our test tube racks hold 6 test tubes, and so I might go up by 2s, 2, 4, 6, 8, 10, maybe 12. I'd have to test it with, 10, you should really already see the drop off. So maybe you do 2, 4, 5, 6, 7, 2, 4, 5, 6, 7, and 10. Or maybe you go do 2, 4, 6, 7, 8, and 10 if you have access to those buffers. Yeah, that's great. That's a great idea. Because you can purchase those pellets that make those. So detailed instructions for the lab. So as much as is possible, we have, we tried to start with the big tasks first. We do not have teacher notes for every single thing. The slides often have notes in the slide notes. So we're trying to add those teacher notes little by little, but our time for that is we try to do as much as we can.